Okay, so let's look at the fluid mosaic model here. The fluid mosaic model. Okay. Now the word fluid here implies what about the cell membrane? What does the word fluid imply? Anyone? What do you think of when you think about fluid mosaic? Good, it's not solid. What else? It's movable, perfect. It's moving and it's not solid. It's not rigid, it's not fixed, all right? And mosaic implies that there are several components that are acting together very nicely to bring about the function that we see. Let's draw another protein here. So what we're drawing here is the phospholipid bilayer to start. And then we have some other interesting components that are in between the phospholipid bilayer here. Okay. First of all, the main component of the cell membrane is the phospholipid. I'll write it here, the phospholipid bilayer. Okay. Then we also have two types of proteins. We have integral proteins. Then we have a protein on the cytosolic surface, which is called a peripheral protein. Okay. We also have the presence of cholesterol here. What type of biomolecule is cholesterol? Anyone? What type of biomolecule is cholesterol? It's a lipid, perfect. Okay, we'll talk about cholesterol here. And then we have some carbohydrates being a part of the membrane as well. When they're attached to proteins, I'll put them over this side. Protein here. So if they're attached to proteins, I'll draw these sort of branch-like structures as carbohydrates. And then if they are attached to a lipid, they are a different type here. So they can be a glycoprotein or a glycolipid, okay? Both of these we will class under carbohydrates. We'll talk about these as well. Let me see that, carbohydrates. Okay, so we have 5 components that we're going to discuss in a little bit more detail as we describe the structure and function of the cell membrane. The first thing we know is that the main structure of the cell membrane is formed by what structures? What structures? What do we call these? We described them and we looked at biomolecules. Phospholipids, right? They have a hydrophilic head and a hydrophobic tail, right? So they are amphipathic, right? They are amphipathic. The head of these structures like water, so they're going to interact with the aqueous environment inside the cell as well as outside the cell. And then the tails are hydrophobic. They do not like to interact with water. Alrighty. So these are going to be forming the main structure of the cell membrane. Also important to note is that they are impermeable to water. Why are they impermeable to water? Why are they impermeable to water and polar molecules? Who wants to talk about that? Why are they impermeable to water? Why is the phospholipid bilayer impermeable to water? I'll zoom this in a little bit here. Come on, we talked about this, Madeline.
The outside is polar and the inside is non-polar.
All right, so the inside is non-polar. So water is not, or polar molecules are not going to be able to traverse this membrane, right? Because of that non-polar or hydrophobic center of the lining of the membrane, okay? Now the phospholipid bilayer also gives a certain amount of flexibility and fluidity to the membrane, right? Flexibility and fluidity. And as you mentioned before, this membrane is not rigid, it's not fixed, it's very movable, and that's going to help in giving a certain amount of function to cellular membranes, be it in the mitochondria, around the cell, and so on and so forth, right? That flexibility or fluidity. Let's talk about integral proteins, right? So the membrane is not just made of these phospholipids. They have other important structures that are embedded within these phospholipids, okay? The first one is integral proteins. And integral proteins are, as the name implies, important, and they are permanent. So they are permanently fixed to the membrane. Okay, now they can span the entire length of the membrane as we see here, or they can just be on one side. Okay, but important is that they're going to be fixed within the membrane. They're going to offer an integral role of structure and support to the membrane. They can be receptors, they can be channels, different types of function, but important to understand is unless this membrane is destroyed or damaged or this cell is lysed, this protein will remain within the phospholipid bilayer. Okay. So let's also make a note here that they are also amphipathic. Amphipathic. And they would have to be amphipathic, wouldn't they? They're interacting with the hydrophobic tails in the center here, as well as the hydrophilic heads of these phospholipids. So by nature, they are amphipathic. They have a polar region and a non-polar region. Alrighty. And as we mentioned before, they can either be one-sided, meaning they span one side of the membrane, or transmembrane. Okay. Alrighty, let's compare those to peripheral proteins. Peripheral proteins. Now these are usually on the cytosolic side of the plasma membrane. So the cytosolic side. These are usually involved, some examples are the signal transduction messengers or the signal transduction receptors that are involved in the signaling cascade for certain processes. We'll talk about those when you look at the autonomic nervous system. They can be receptors as well, channels, and different types of things. But the important part is these can easily dissociate off. They can easily dissociate. So they can be present for a certain function, they can degrade away, they can be recycled, and then they can be repaired or returned to the membrane. Okay, so they are not permanent. Also important to note is that they are anchored or tethered to the cytoskeletal elements of the cell. Okay, does anyone know what the cytoskeletal elements of the cell are? What do we mean by cytoskeletal elements? Come on, this should be a review for you guys. We're talking about some of the basic organelles of the cell. What are cytoskeletal elements? What is their role in the cell? These are your microtubules, your filamentous proteins, right? They help to anchor the cell. They give the cell shape and structure, almost like a scaffolding network within the cell, okay? And so they anchor the peripheral proteins to the cytosolic side of the cell membrane. Any questions so far? Any questions so far? Let's move on and talk about cholesterol, right? Cholesterol. What type of biomolecule is cholesterol? What type of biomolecule are we talking about with cholesterol? We said it's a lipid, perfect. Okay, and it's going to offer some additional flexibility and fluidity. Okay, and it does so by breaking up some of the hydrophobic interaction between the fatty acid tails. So I mentioned that here, breaking fatty acid interactions. Okay, so those fatty acid tails can have a propensity to want to stick together, to sort of crystallize, to form this rigid structure. And having cholesterol there helps to break up those hydrophobic interactions and help to offer some additional flexibility, fluidity to the cell membrane, which is very important. Okay, so they're going to decrease crystallization, and decrease rigidity. They are also impermeable to water, so they are a lipid, okay? They are non-polar. So they offer this, this integrity to the membrane where it's not leaking. Water cannot easily move through the membrane. We don't want intracellular fluid sort of just leaking out of the cell through the membrane. And we don't want extracellular fluid leaking through the cell as well. Okay, so having cholesterol here helps to prevent that movement of water through the cell membrane. Okay, so we're going to say decrease leaking, right, or impermeable to water. Okay, and these are some of the features that cholesterol offers to the cell membrane. Let's talk about carbohydrates lastly. Let's talk about carbohydrates. Now, if you see the prefix glyco, we know that a carbohydrate is being added here. So hence glycolipid, we have a carbohydrate attached to a lipid. And glycoprotein, we have a carbohydrate attached to a protein. Okay. Now, carbohydrates help with cell tagging. They're important in cell identification, so cells being able to signal or identify themselves. They're important in the ABO blood grouping or the blood typing. That's how you know what blood type you have. They're also important in the immune system. So certain antigens or certain immune signals are being presented using these glycoproteins. Ready? And then they are covalently bound to either the protein or a lipid. So there's a covalent bond between that carbohydrate and either a protein, making it a glycoprotein, or a lipid, making it a glycolipid. Okay. I'll zoom out a little bit. So these are some of the features of the cell membrane. These help the cell membrane to transport certain substances and particles across, but it also maintains a barrier. Okay, very unique environments on either side because of the selective permeability of this membrane. We'll talk about that in a minute. But what I really want you to understand here is because of these unique features, we got cholesterol, we have the proteins, and most importantly, we have the amphipathic nature of the phospholipid bilayer. We have the compartmentalization of the cell, where we have a unique environment happening inside the intracellular space, and we have the unique environment outside the extracellular fluid compartment or space. Any questions on